Hello everyone, welcome back to our YouTube channel AgriAddict, a platform for all agricultural exams. This is a free lecture series for IBPS AFO mains exam and also useful for other agricultural exams. I am your mentor Thrilok Srinivas. I completed my undergraduation in Agriculture College Bapatala in Acharya Engineering Agricultural University, Andhra Pradesh. Currently, I am pursuing my masters in UAS Dharwad in the Department of Genetics and Plant Breeding. I secured an All India rank of 25 in this year's JRF Plant Sciences. So without any delay, let's get our video started. Our today's topic is Mendelian Genetics Part 2. I hope you all understood and liked the Part 1. So in this Part 2, we're going to learn about gene interaction, types of gene interaction, qualitative and quantitative characters, multiple factor hypothesis, expressivity and penetrance, cytoplasmic inheritance, and maternal effect. Let's know what is gene interaction. Gene interaction means two or more genes affecting the expression of each other. Means let us take two genes like A and B, and if character of A is being affected by the B gene or the character of the B, or character of the B is being affected by the A gene or the character of A, we call this as gene interaction. And here we have three terms like epistasis, epistatic gene, and hypostatic gene. Let's know what are these terms. Epistasis. Epistasis means a phenomena where expression of one gene is modified by the expression of another gene. Means it's nothing but if the expression of A is modified by the expression of B, we call it as epistasis. And due to B, the A's character is modifying, so we call B as epistatic gene, and the A is being modified or suppressed, so we call A as hypostatic gene. Means a gene which suppresses or masks called as epistatic gene, which is being suppressed we call it as hypostatic gene. And these are the types of gene interactions. Let us know one by one in detail. Here first one is independent assortment. Independent assortment means nothing but no gene interaction will be present. Here both the genes are independent and they express their characters independently. Here you can see this is the independent assortment, and the phenotypic ratio is 9:3:3:1. We can also explain this 9:3:3:1 as 9 means when both the alleles are in dominant state, the phenotypic expression will be 9 out of 16. Means in 16 plants, 9 will be having both the genes as dominant. And 3 means one gene is dominant, Y or R. And 1 means both are in recessive condition. Like this we can remember. And the first type of epistasis is dominant epistasis. Dominant epistasis means first gene, when dominant, masks the expression of second gene. Simple. Means when A is dominant, it will mask the expression of B. So here we can get A. Here also the small b will be masked. So we will get A. Here A is in recessive condition, so it cannot mask the B. So we get B. Here also we get small a and small b. Small a cannot mask the expression of B, so we will get B. So if we take this as phenotypic ratio, we can get A plus A means 9 plus 3 as 12 as phenotypic ratio. Here you can get 3, here 1. Like this, we get the phenotypic ratio of 12:3:1. So if you understand the definition correctly, you can get that ratio accordingly. Next epistasis is supplementary epistasis. In the supplementary epistasis, there will be two conditions. First condition is second gene has no expression, and when it is present with first gene, it modifies the expression of first gene. This is the first condition. Second gene has no expression, and when it is in first gene, it modifies the expression of first gene. Whereas second condition is first gene is if recessive or both the genes are in recessive, then no character will be expressed. Suppose here B is the second gene, A is the first gene. Here B is in dominant state. So A will be modified as A dash. Here B is in recessive state. So A will be expressed as A. Here B is in dominant state, but first gene is in recessive condition. So no expression. Here also first gene is in recessive condition. So no expression. Now we can get this as A dash as 9, A as 3, and the none as 3 plus 1 is equal to 4. Like this, we will get 9 is to 3 is to 4. Next one is complementary epistasis. Here you can get 9 is to 7. How? We will discuss. Complementary epistasis means both the genes are in dominant condition to produce dominant trait. If one or both genes are in recessive state, then the contrasting trait will be expressed. Means here both are in dominant, so dominant will be expressed. Here only A is dominant, so it will be expressed as recessive trait. Here only one gene is dominant, so dominant trait will not be expressed. Here also only one gene is in dominant trait, so recessive trait will be expressed. Here both are in recessive, so obviously recessive trait will be expressed. Means in the complementary epistasis, if there are two genes should be in dominant condition to produce dominant trait. If one or both the genes are in recessive, then the expressed character will be recessive. Like that, we can get here as dominant is to recessive as the phenotypic ratio. Dominant means 9. Recessive means 3 plus 3 plus 1 is to 7. And the next one is duplicate gene interaction. Duplicate gene interaction means nothing but two genes are of same character. But when the both genes are in recessive state, then other character will be produced. Suppose here A and B produce same character. So it will be produced as, let's assume it as T. Here A is in dominant state. So again T will be produced. Here also B is in dominant state. So again D will be produced. Here A and B both are in recessive state. So recessive character will be produced. If you take this as phenotypic ratio, we get as 9 plus 3 plus 3 is to 1 is equal to 15 is to 1, because both alleles control same character. So if either one is in dominant, then it will be enough to express the character. That's why we call it as duplicate gene, because both are expressing same character. The next one is inhibitory epistasis. Inhibitory epistasis means one gene expresses the trait when in dominant stage, but if second gene is also in dominant state, it inhibits the expression of first gene, irrespective of its genotype. You got it? Means first gene is in dominant state, but if second gene is also in dominant state. Okay, let me explain clearly. Here, this is first gene, this is second gene. If second gene is in dominant state, it will inhibit the first gene, so no expression. Here second gene is in recessive state, so first gene will be expressed, capital A. Here second gene is in recessive state, so no expression. Here both are in recessive state, so no expression. Means if B is dominant, then there will be no expression, because if A is recessive or dominant, no matter, because B inhibits A. That's why it doesn't matter whether A is dominant or recessive, there will be no expression. So I think you got it. Here the phenotypic ratio will be 9 plus 3 plus 1 is to 3 is equal to 13 is to 3. And this is the last gene interaction, which is additive gene interaction. Additive gene interaction means both the genes express same character when they are dominant singly, But when both are in dominant condition, expression will be enhanced. Means A and B, same character will be produced. But when both A comma B are in dominant condition, expression will be developed. Like if A produce 1C, B produce 1C, when both A and B are in dominant stage, then it will be produced as 2C. Here you can see both are in dominant stage, so it will be 2C. Here 1C, here also 1C, here it will be 0C. So we get the phenotypic expression as 9:3 plus 3:1, which is 9:6:1. That's it. Like this, if you understand the definition, you can get the ratio as correctly. So I hope you take this table as screenshot and refer it later. So the next topic is quantitative and qualitative characters. So what are quantitative characters and what are qualitative characters? Quantitative characters means which can be measurable, like weight of the seed. Weight of the seed is measurable. We can measure it on the weighing machine. And height of the plant, we can measure it using scale. Like that, the measurable ability of a character, we call it as quantitative. If it is non-measurable, like color of the seed, we cannot measure the color. Like if it can be yellow or green or something else, we cannot measure the color. That we call it as qualitative character. And quantitative characters are controlled by polygenes, means many genes control single character. Here oligogenes means only few genes will control the character. And the effect of each gene will be low, because as there are many genes, suppose 10 genes are there, then each will get only 10% effect on the character, because it is a polygenic. Whereas in oligogenic, let us assume like 2 or 3, then the effect will be 50%, like that. Means for each gene the effect will be high in qualitative, it will be low in quantitative. And the effect of environment, here you can see like weight of the seed and height of the plant, like these characters, these are the highly influenced by the environment. Whereas the color of the seed, it doesn't change whether the environment is favorable or non-favorable. It will be same regarding the environmental character, means it does not depend on the environment. And the frequency distribution in the population. Frequency distribution means like if we have to take a table, like number of plants are yellow, green, we can take it as 12, 15, like that in a discrete number. Whereas of the quantitative character, like weight of the seed, some can be 2 grams, some can be 2.5, some can be 3 grams. So like this it will be continuous. So how we will be take this? We will divide them into classes, like 0 to 2, 2 to 4, like that we will divide. This we call it as continuous data, means this is not discrete, and this should be taken as the frequency distribution table as a continuous data. Whereas here it will be discrete, yellow or red, that's it. But here continuous means we have to measure the weight, like 2 grams, 2.1 gram, 2.2 gram, like that. And the graph also same, it will be expressed as continuous curve, it will be expressed as discrete classes or bars, like this, classes or bars. And we can study this quantitative character using mean, variance and statistics. Like we will take the mean to get the average weight of the seed, like 2, 2.1, 2.2. We will take this average and we will take it as 2.1. But here qualitative character, we cannot take the average of the yellow and red. We will take the ratio, like how many are yellow, how many are red. We will take the ratio. And the transgressive segregation. First, let us remember that in the quantitative character, transgressive segregation is present, in the qualitative it is absent. In the next slide, I will tell you what is transgressive segregation. Next one is multiple factor hypothesis. Multiple factor hypothesis, this is the important bit, like who postulated it? Yule. Yule postulated it in 1906. But the experimental evidence, means the proof that multiple factor hypothesis is true, it is provided by Nielsen et al. in 1908. So what is multiple factor hypothesis? Multiple factor hypothesis means, let us assume there are 4 genes, 1, 2, 3, 4. These will control the character. Like, let us assume weight, 10 grams of the seed. If these 4 genes are present in dominant, 1, 2, 3 and 4, these 4 are produced in dominant, then it will be 10 grams. But if only 1 is in dominant, like, okay, let's assume 3 are dominant, 1, 2, 3, then 7.5 grams. If 2 are dominant, 1, 2, 5 grams. If 1 is dominant, 2.5 grams. Like this, many genes, each with a small effect, contribute to genetic variation. Here you can see, there are many genes. Each gene is contributing something. Like first gene is contributing 2.5, second gene is contributing 5, third gene is contributing another 2.5. Like that, each gene is having small effect on the same character. This you have to remember. Many genes with a small effect which contributes to genetic variation. This is the multiple factor hypothesis. And we can also call it as polygenic inheritance. Remember this. Here you can see, in this condition, 4 are in dominant stage. So there is a dark red. Here 4 are in recessive state. So there is white. But in the hybrid, only 2 are in dominant. So it is medium, intermediate expression. Here only 3 are in dominant, means medium dark red. You can see, based on the number of the dominant genes, the character expressed is changing. So it means each gene has its own role in the character, like R1, R1, R2, R2. In this, first gene, second gene, third and fourth. These 4 alleles have some effect on the character. And the next one is transgressive segregation, means formation of extreme phenotypes observed in segregated hybrid populations compared to phenotype observed in parental lines. Let me explain. Here you can see, in one parent the score is 0, in the other parent the score is 0, but when the F1 is crossed or selfed, we can get the score as +4, -4, +2, -2, etc. Means the progeny are having more extreme characters than the parents. Progeny greater expression than parents. This we call it as transgressive segregation. And this is the principle we use in heterosis also. Right? If the parent, you can see why it is getting the more expression, because one parent is having dominant in one allele, other parent is having dominant in other allele. So when these two are crossed, hybrid is having both the alleles as dominant. So if we self this, we get some extreme phenotypes which can be used for our purpose. Like this, transgressive segregation is seen in the genes which are controlling multiple factor hypothesis or polygenic inheritance. We cannot see transgressive segregation in normal genes, but genes which are polygenes or quantitative characters, in that we can see transgressive segregation. Okay, transgressive segregation means progeny are having more extreme phenotype than parent. Remember this, simple. And these are some of the important terms you have to learn. Major genes. Major genes means few genes with large effects, means Some genes, they may be few in number, but they control majority of the characters. Then we call it as major genes. You can see few genes with large effect that influence most of the genetic variation. For suppose like qualitative characters, like color of the seed is controlled by only few genes, like 1 or 2. Then only these 2 genes are producing some major change. So these we call it as major genes. Whereas minor genes means, let us take quantitative character like weight of the seed. It is controlled by, for example, many genes like 10 to 12 genes. Then each gene will show low effect or small effect. So we call it as minor gene. Means there will be many genes, but the effect will be small, that influencing the genetic variation. So we call these as polygenes because many genes are showing the influence in small effects. And the next one is quasi-quantitative characters. Means the characters will be governed by the polygenes, but we cannot express them in discrete way. Continuous variation. Means quasi-quantitative characters means these are controlled by polygenes. What we learned in the previous slide, if a character is controlled by polygenes, it will come under quantitative. But even though these characters are controlled by polygenes, we cannot say them as quantitative because they cannot be expressed in continuous. Continuous means like 1, 1.1, 1.2, like this, we cannot express this character. So such characters, we call it as quasi-quantitative characters. You can see here, like number of branches per plant, number of pods per plant, like that. Even though these are governed by polygenes, we cannot express them as 1.1 branch, 1.2 branch, 1.7 branch, like that. No, we will express them as 1 branch, 2 branch, 3 branch. So these we will call it as quasi-quantitative characters. And the next one is quantitative trait loci. Quantitative trait loci means the locus or the site of the chromosome at which genes affecting a quantitative trait is involved. Means we all know these are the polygenes. Polygenes affect a character, for example, weight of the seed. This is a chromosome. If the weight of the seed is governed by 10 genes, these 10 genes are present in this loci. Then this loci we call it as QTL, quantitative trait loci. Means the position or the locus in which the genes which control quantitative characters are present. We call it as QTL. Means the genes which present or which control quantitative trait are located. They call it as QTL. Understood? And these are some other terms like phenocopy effect. Phenocopy effect means in some environments, the expression of gene will be such that the resulting phenotype is same as produced by the another allele. Then we call it as phenocopy. Means this is the genotype of the plant. Means the plant should show tall. For example, let us assume that the plant should show tall. But if the plant is lacking no nutrition, no water, no sunlight, then the plant will show dwarf. Even though the genotype should represent tall, but according to the environment, it will be dwarf only. Means the environment is changing the expression of a gene so that the phenotype will be differing from capital A into small a. So this type of environment or the effect due to environment on the phenotype, we call it as phenocopy. And the next one is expressivity. Expressivity means the degree to which a genotype is expressed as a phenotype in an individual. Here there are two types of expressivity: uniform expressivity and variable expressivity. First of all, let us learn what is expressivity. Expressivity means, here you can see in this, let us assume these are plants 1, 2, 3, 4, 5 and 6. In these 6 plants, the expression of the character is same. All are expressing complete green color. So we call this as uniform expressivity, means the gene or the character is expressed in all individuals in same manner. So we call this as uniform expressivity. Whereas variable expressivity. Variable expressivity means the gene's phenotypic expression differs. Means if in one plant 100% it is expressed, in another plant 90% will be expressed, in some other plant 60% will be expressed. Like this, the expression of the character will be varied. Then we call it as variable expressivity. And the next one is penetrance. Penetrance means the frequency or percentage with which a gene produces its visible effect in individuals who carry it. For example, this is the example of the penetrance. Here you can see in the 6 plants, let us assume capital A is the genotype. Here the character is expressed in all the plants. The capital A's character expressed in all the plants. But here you can see only in some of the plants the character is expressed, but in others it is not expressed. Then we call it as incomplete penetrance. Means the percentage in which the gene shows its effect. It is known as penetrance. Complete means 100% the gene will be express itself in every individual. But incomplete means a condition where genes express themselves in only some but not in others. Like you can see here, in all individuals the genotype is not expressed. Only some of the individuals the genotype is expressed. So we call this as incomplete penetrance. Here in every genotype or every individual the character is expressed. So we call it as complete penetrance. And narrow expressivity. Narrow expressivity means every narrow or uniform expressivity. Uniform expressivity means the expression of the gene is same in all individuals. But variable expressivity means the expression differs. In some it can be 100, in some it can be 70, or in some it can be 60, like that. The next topic is cytoplasmic inheritance and nuclear inheritance. So what's the difference? Like, what is cytoplasmic inheritance or what is nuclear inheritance? Cytoplasmic inheritance is the term which is derived because we all know that in nucleus DNA will be present, and we also know that other than nucleus some of the DNA is present in cell organelles like mitochondria, chloroplast. Like this, in some cell organelles the DNA will be present. So you all know that DNA will be in turn as gene, as chromosome, etc. Like that, samely in this DNA also there will be some genes which control some characters. Transfer of the genes from the cell organelles from parent to progeny, we call it as cytoplasmic inheritance. Means inheritance of genes present in cytoplasmic organelles. That's it. Nuclear inheritance means inheritance of genes present on the chromosomes. And mediated means the cytoplasmic inheritance, the genes are transferred mainly from maternal, means from the mother's side, whereas the nuclear inheritance, both maternal and paternal, means both the parents are involved in the inheritance or the transfer of the genes. And the location of the genes, here the location of the genes is in mitochondria and chloroplast, whereas here it is the chromosomal DNA which is located in the nucleus. And Mendelian inheritance, which follow the Mendel laws. As you all know, nuclear inheritance follows the Mendel laws, like 3:1 ratio, 1:2:1 ratio, like this. Nuclear inheritance follows the Mendel's laws. But cytoplasmic inheritance, it does not follow the Mendel's laws. Let me show you. If this is the mother cell or paternal cell, in the progeny, all will be expressing characters of the mother. In which condition? In cytoplasmic inheritance condition. But what is the Mendel's laws? Both mothers and father's condition should also be expressed in the progeny. Then we call it as Mendelian inheritance. But here only maternal or the maternal characters are expressed. So this won't follow Mendelian inheritance. Whereas nuclear inheritance follows the Mendelian inheritance. And the number of genes transferred. As you all know, mitochondria and chloroplast has only small amount of DNA, so small number of genes only transferred, whereas majority of the genes are nuclear inheritance, so there will be many genes. These are the main differences about cytoplasmic inheritance and nuclear inheritance. So knowing these differences will be enough. There will be nothing to see deep in the topic. And the next one is maternal effect. What is maternal effect? Means the phenotype of the progeny is influenced by genotype and environment of its mother. Means, let us assume mother and progeny. The characters of progeny influenced by mother, either due to mother's genotype or the mother's environmental condition, the phenotype of the progeny will be influenced. Example, during the oogenesis or the formation of the egg cells, the mother's gene expression influences the development of the embryo. Right? If the mother's gene expression do not favor oogenesis, then there will be no embryo development. If the gene expression favors the embryogenesis, then there will be the development of the embryo. Simple. That's it for this part. In the next part, we will know about the linkage and crossing over, types of linkage and crossing over, coincidence and interference, and the mechanisms of the sex determination. As you all know, AgriAddict launched a new course, Vijeta. This is a useful course for the IBPS AFO MAINS exam 2024 because it contains 50+ standard mock tests, previous year papers and solutions, one-to-one doubt clearing sessions, and the PPTs which we describe in this video, all will be present in this program, IBPS Vijeta. So, those who are preparing for IBPS AFO mains exam, I suggest you to take this program and get a good ranking for this exam. That's it for today. Thank you. Let us see in the next part, Mendelian genetics part 3. And one more thing is, if you have any doubts regarding the Mendelian genetics, please post your queries in the comment section. We will see your comments and solve your doubts as soon as possible.